Welcome, and thank you for taking the time to watch this video on Transcelerate's initiative to address industry-wide problems related to regulatory guidance on protocol deviations. Protocol deviation, or PD management processes, are complex, varied, and broadly impacted by a wide range of definitions for identification and classification. These definition differences are associated with process inefficiencies for both sites and sponsors that can hinder rapid identification of important protocol deviations. Transcelerate launched an initiative to address industry-wide problems related to interpretation of regulatory guidance on protocol deviations. My name is Laura Ramos Castillo, Business Process Owner for Protocol Deviations at Pfizer. I've been working in clinical research for 20 years with a focus on the protocol deviation space for the last 7 years. I'm currently the co-lead of the Transcelerate Protocol Deviations workstream. Managing protocol deviations is complicated. The lack of clarity regarding the definition of important protocol deviations leads to both over and under interpretation of deviations, which impacts the planning, collection, analysis, and reporting of important protocol deviations. This over and under interpretation could increase noise in the protocol deviations management process, potentially hindering the identification of important participant safety information and influencing the reliability of trial results and human subject protection. One example of overinterpretation may be classifying every required missed diary entry as an important protocol deviation when there's no impact to the study's endpoints or the participant's safety. Classifying these protocol deviations as important in this case could create noise and make it more challenging to detect true PD trends and signals. Alternatively, underinterpretation of an important PD may also have a negative impact. For example, a team may assess individual PDs related to a missed procedure, such as a rater assessment, as a non-important PD. However, if the missed procedures reach a certain threshold on the study where there's insufficient data to assess an endpoint, there could be downstream implications about the use of that data and its reliability. With feedback from stakeholders, including health authorities, the workstream developed a toolkit to support clarity in definition and a holistic approach to the management of protocol deviations. Improved protocol deviation processes should ultimately lead to improved participant safety, reliability of study data, human subjects protection and data quality, and more rapid identification of protocol deviations. There are potential benefits for participants, sites, sponsors, and regulators. The goal of the Transcelerate Protocol Deviation Initiative is to support rapid identification of important protocol deviations and increase participant safety and process efficiencies for sites, sponsors, and regulators. The proposed methodology to accomplish this includes improving interpretation of guidelines, including definitions, improving management of protocol deviation processes, and engaging with health authorities on these critical issues and solutions. This video will walk you through the different components of the Protocol Deviations Toolkit, which was designed to make the protocol deviation management processes more efficient. We will also take a look at some examples of how the toolkit can be used in practice. The PD Toolkit is comprised of the following 4 components. The PD Process Guide, which is a framework describing flexible PD management approaches based upon a proposed interpretation of the ICH E3 definition for important protocol deviations and other associated PD guidance. The PD Map, which is a visual representation of the end-to-end PD management process containing pathways for both important and non-important PDs. A Protocol Deviation Assessment Plan, or PDAP, which is an Excel template to help teams prospectively identify and document classification of importance, categorization, responsible roles, and other elements related to PDs. and a decision tree, which helps users determine if an event is a protocol deviation, and if so, whether it should be classified as important or non-important. Application of the components is not a one-time event at the beginning of the study. It's anticipated that these materials will be reviewed and refreshed to reflect real-life situations and decisions during the conduct of the study. Feedback loops and other mechanisms are noted within these tools. We will now do a few demonstrations of how the elements of the toolkit work. Let's start by orienting ourselves on the end-to-end process by using the Protocol Deviations process map. You can access the map on the Transcelerate website by navigating to the Protocol Deviations Solutions page. Once there, starting on the left side of the map, you'll see that the process begins with the Prepare step. This first step is to define and prospectively identify important protocol deviations which may occur. Inputs may come from organizational, intermediate, and protocol level components as illustrated in the blue triangle. Although all three inputs are used, we note that organizational and intermediate level definitions of importance are used whenever possible, augmented with protocol level definitions where necessary. This will reduce variability in classification and categorization and help to support consistency in analysis and reporting within the department, company, or organization. These inputs, such as company or organizational policies or SOPs, the protocol, statistical analysis plan, and other study plans can be used to create what we call a Protocol Deviation Assessment Plan, or PDAP. Let's imagine for a moment that you are a member of a study team who would like to implement a PDAP for the first time. You start by reviewing your policies and SOPs and see that your organization considers all protocol deviations that deviate from key eligibility criteria to be important, in alignment with ICHE 3 guidance. You decide to use the Transcelerate PDAP as your starting point, so you navigate to the Protocol Deviation Solutions page on the Transcelerate website and download the Excel PDAP template. After reviewing the Overview and Instructions for Use tabs, you're ready to begin drafting the PDAP for your study. You decide to start with the organizational level PD related to key eligibility criteria. If your organization uses the same PD categories as the Transcelerate initiatives, you can use the drop-down menu for category. If not, simply add in your organization-specific options. For PD description, you enter your organization's specific language. For identification method, use the drop-down menu to indicate how you would expect to identify this type of protocol deviation. Enter the role responsible for identification of that type of PD. Both columns G and H refer to the classification of importance based on the impact to the data and the impact to participants, respectively. If the PD is not considered important, indicate whether if it reached a certain threshold, it would be classified as important and define that threshold in column I. Otherwise, select No. After completing all of the respective protocol deviations identified at the organizational, intermediate, and protocol levels, and documenting the version history, the document is ready for study team review and finalization. In another scenario, imagine that you already have a PDAP equivalent for your ongoing study, but you have been tasked with adding information in your study's monitoring plan to clarify the expectations of how PDs will be identified and what should be considered a PD versus monitoring issue. After consulting the PD process map, you see that under the Identify and Collect section, there are two methods of identifying PDs described: programmatic identification and manual identification. You review the PD process guide and decide that you will include a description and examples of each in your study's monitoring plan. Programmatically identified PDs are based on data captured in a database using an electronic computerized process. For your study, you provide the example of required site visits performed outside of the protocol allowable window, as these will be identified through data listings. Manual identification relies on human interpretation, so you note that an example would be informed consent deviations that monitors may uncover. Continuing on in the scenario, imagine you have also been asked to provide additional information regarding what should be considered a protocol deviation in the study monitoring plan. So you consult the Transcelerate clarifying principles to identify protocol deviations and the protocol deviation decision tree. You note that to qualify as a PD, 1. The event has to have occurred to avoid theoretical situations. 2. That the event is related to the protocol or documents referenced in the protocol. And 3. The event is independent of fault, blame, or circumstance to ensure an objective approach to identification. You list a few examples, such as a lab sample that was collected by the site but lost en route to the central laboratory, a participant who missed a visit because the site was closed due to inclement weather, and a participant who refused to complete a questionnaire. These are all protocol deviations because they relate to a data point or process required by the protocol that was not obtained. You also decide to provide a few examples of issues that are not protocol deviations, such as correspondence that was not in the study binder at the time of the monitoring visit but was captured, and a participant who signed the informed consent document below the designated line, since these can be easily addressed via routine monitoring follow-up. The documents and tools contained within the PD Toolkit will require careful thought and will take some effort the first time they're created. However, we have received feedback that the tools can be leveraged for reuse on future studies for the same investigational medicinal product or within the same indication, which reduces effort the next time around. Now that we've shared some of the different components of the PD Toolkit, you can see that the approaches described were designed to be flexible to suit a variety of indications, study designs, and investigational agents, while also providing tools for consistent application within a study, program, or organization. They can be applied and adjusted based on your organizational and study-specific needs. Individual components of the toolkit can also be adopted in any order. It is recommended that you assess your PD management processes in an effort to increase efficiencies and help reduce noise to support rapid identification of important PDs, thereby focusing on what really matters. All of our tools and solutions can be found on the Protocol Deviations page in the Sponsors section on Transcelerate's website. This concludes our presentation. Thank you for watching, and good luck with your future clinical research endeavors.